Hey everyone, in this video we're going to talk about proteins and nucleic acids, and then we'll be done with this chapter. So proteins are organic molecules that contain carbon, hydrogen, and oxygen, which we've been talking about. Plus, in this case, we're going to have nitrogen added to the group and sulfur to some of the proteins. So proteins have multiple functions. Proteins can be enzymes. Enzymes are structures that increase reaction rates. Proteins can be hormones, which tell cells what to do. Proteins can be antibodies, which protect. Proteins can build up structures, so they can be structural. And so I give the example of hair and skin. So proteins have a lot of different jobs. The building blocks of proteins, so these are the polymers, are amino acids. Amino acids are monomers. There are 20 amino acids that make up every single protein. So we have a total of 20 amino acids, and every protein on Earth is made up of those same 20 amino acids. How crazy is that, right? So amino acids contain a central carbon. Then we have an amine region over here, a carboxyl region over here. We have a hydrogen bond, and then we have this R group. The R group is the unique structure that makes each amino acid different. So there are 20 different R groups that make the 20 different amino acids. And like I said, there are 20 different amino acids. They make up every single protein. And, just off the top of my head, skin is protein. Muscle is protein. Blood is protein. Bone has protein. Connective tissue is protein. So tendons are protein. So, that's just off the top of my head. And those are all, well, not all, but most of those are structural proteins. And like I said, we have amino acids, or we have proteins that are enzymes. We have proteins that protect us. So we have a lot of different types of proteins, thousands of different proteins. Proteins tend to be produced when more than 100 amino acids come together, and the shape is important to for the function of the protein. I always tell my students, structure determines function. So the structure is going to determine how the protein works. And that is true. That is true in everything. I teach this in anatomy, but it's true outside of that class. It's just true. Structure determines function of everything. If a straw, the structure is flawed, there's a hole in it, you're not going to be able to sip milk or soda or juice through that straw. It's not going to work because the structure is flawed, and so the function will be flawed. So proteins have 4 different structures. So the first structure is called a primary structure, where we have just a bunch of bonded amino acids. The secondary structure occurs when you have hydrogen bonds between one amino acid within the protein and another amino acid. Tertiary structure occurs when you now have covalent bonds along with those hydrogen bonds and ionic bonds. And then some proteins have that fourth structure, which is quaternary. So let's look at these. Here's that initial primary structure of a protein. We just have multiple amino acids bonded together. The secondary structure then occurs when you have hydrogen bonds that are pulling the protein into a different form. So you can have two different types of secondary structure. You can have the alpha helix or a beta pleated sheet. So these are secondary. Tertiary structure occurs when the protein folds up into its structural or globular shape. So it's going to fold up into its functional form, and then it can do its job, okay? And so here you have alpha helix, here you have beta pleated sheet, and the protein folds up all together, and now it's able to work. A quaternary structure occurs when you have two or more proteins that fold together. So you get two or more of these structures here that start folding, and they fold all together, forming a larger structure. So in this case, we have two proteins that are identical and two other proteins that are identical. And this actually is, and it's probably the most famous form of a quaternary structure, is hemoglobin. Hemoglobin is a protein that's found in blood, and it's a quaternary structure. It's formed by having multiple primary or primary proteins folded together. So proteins can lose their structure. So if a protein loses its structure, then its function changes. So here is an egg, and I have that as the example here, so I took this picture. Here's an egg on a frying pan, and the initial structure of the egg, you have the yolk, and then you have that albumin structure. Albumin is this mucousy clear material, but as we change its structure, it turns into this rubbery white material. It loses its function. It can no longer do its job. And then over here, the protein is pretty much completely denatured, and now its job would be to be eaten, right? So in our cells, the same thing happens. If we have proteins that denature because of, say, body temperature change, that can actually lead to brain damage because, again, like I said, everything is made of protein. So our brain is composed of proteins. So if our proteins get heated up too much, or something else changes, like pH changes, that can actually cause denaturation, and that can lead to brain damage and death of cells. The last structure that I'm going to talk about, or the last organic molecule I'll talk about, are nucleic acids. Nucleic acids include your DNA, your RNA, and ATP. Nucleotides are what make up nucleic acids. And so over here are the different bases. We have adenine, guanine, cytosine, thymine, and uracil that make up these different nucleotides, which make up the nucleic acids. So DNA is probably the most famous of all of our nucleic acids. DNA is what contains genetic information that makes our proteins. So it carries information for protein synthesis. It's also what we pass from parent to offspring. So it's the information that's passed from one generation to the next. In DNA, the bases are adenine, thymine, cytosine, and guanine. Adenine and thymine always pair together. Cytosine and guanine always pair together. The sugar in DNA is deoxyribose, and DNA's backbone then is a sugar phosphate backbone, and it's found in a helical form. So this is what DNA looks like. Here's the backbone. You have sugar, phosphate, sugar, phosphate, sugar. And on each of these sugars, we have different nucleotides. So A, T, G, C, like I said. And then they're bonded together. So adenine and thymine always bind together. They have a complementary shape. Cytosine and guanine, again, are bonded together. They have complementary shape. Ribonucleic acid, or RNA, then, is the structure that carries out the information, the job that the DNA codes for. So it carries out protein synthesis. There are different types of RNA, but all RNA is single-stranded, contains the sugar ribose, and contains adenine, which is spelled wrong there, but that's okay, guanine, cytosine, and uracil. So there's no thymine in RNA. Instead of thymine, we have uracil, okay? And then we have our ATP. ATP is the energy currency that our cells use. ATP is composed of adenine, which is here, ribose sugar, and 3 phosphate groups. That's what makes ATP, adenosine triphosphate. So ATP then releases energy when it breaks the bond between the second and third phosphate. So right here, this bond is broken. We produce an adenosine diphosphate, and we have this inorganic phosphate that's there. So we break that bond, produce adenosine diphosphate. But if we need to build up ATP, we can add another phosphate to an ADP, producing more ATP as well. That's the end of our video. So I will post this and talk to you later. You guys have a wonderful day. Bye.